Thank you, Beijing. Thank you for the introduction. I'm very happy to be here today. Judith and I will be presenting the ISPE guide on equipment design and qualification of cellular products, which we were both co-authors of. And just to give a little bit of insight into myself after that great introduction, I work for an architectural engineering firm, so my viewpoint from this is supporting the clients that I work with in their technology selection and facility design approaches. Judith?
Thank you so much, Peyton and Stephen, and thank you everyone for joining us. We are definitely excited to tell you a little bit about our findings and about the work that we put in with the larger group, Stephen and I just representing here on the equipment design. Also in my daily work, I'm focusing a lot on how to improve cell therapy processes, more from a digitization aspect, of course, when it comes to how can we do the digital transformation, what are specific challenges in cell and gene therapies, specifically in the current state of the industry in comparison to traditional pharma. And I'm very lucky to be able to be working in the ISPE with different groups, but also with other societies. And I think whenever we can come together and discuss how we can improve the industry that we are invested in and focusing on, it's always a great discussion. So Paisley already posted it in the chat. Please feel free to post any questions in between. We don't need to wait until the end. We definitely would like to have this an engaged discussion, and also if maybe we are saying something and you have a different perspective or you would like to comment, please feel free to do so, because obviously this is a collaborative effort. So yeah, I'm looking forward to the session.
Thanks, Judith. So I will kick off the presentation. So just want to give an introduction to what the guide is initially. So cell therapy, cellular therapy products, the focus is on cellular therapies. There are unique challenges associated with these types of therapies compared to traditional biopharmaceutical manufacturing modalities. And since the first commercialization of CAR T therapy back in 2017, the industry has been evolving rapidly. And therefore there's been a lack of consistency and a race to get the therapies to market has created a lack of harmonization and a lack of coordination between the different companies involved. So really, this guide has been developed with an aim to improve that, with the aim to address that, to look at the different aspects associated with the industry, from production to quality to digitalization, automation, analytics, and looking at how the industry is progressing. So one of the things to highlight about this guide that is a little bit different than any of the other ISP guides that you might read is that there is a significant amount of process equipment actually listed in this guide. So, and that equipment is the manufacturers and the models are highlighted for each of those pieces of equipment. We had a good discussion when we were developing the guide on this, and it was felt that we couldn't write a guide on cell therapy equipment without actually discussing the equipment. So this is why we have taken this approach, and the reason that the comprehensive listing of equipment is included is because the equipment is quite unique to this particular industry sector. So we can't talk about, say, bioreactors in general terms. We need to talk about specific equipment because the equipment is quite specific to the specific unit operations associated with this sector. One thing I would like to highlight, though, is that the information provided is for general information only to guide the reader's decision making process when it comes to the approaches that you would like to take. There are no recommendations in this guide with respect to which technology should be used with respect to which step. The information is there to guide the reader's decision-making process to select the most appropriate technologies for your particular application. And just as I mentioned, the industry as a whole is evolving very quickly, and so the point in time, the guide had to be stopped at a point in time in order to publish it. So we have a section which is discussing future-looking technologies, which really focus around the more roboticized technologies, and highlighting that this is where the industry is going, but without any real focus on those technologies. They will potentially be looked at in more detail at a future revision of the guide, but for this time they're just looked at from a general discussion point. So, and then just an overview of how the guide is structured. So chapter 1 gives you a general overview of the purpose and objectives of the guide. Chapter 2, which I'll discuss now in a bit more detail, gives some general guiding principles towards process, GMP, contamination control, etc. Chapter 3 is a more detailed look at the specific process technologies. So all the different line items here that you can see on the chapter 3 outline the different process steps associated with typical cell therapy processes, and this section of the guide, which Judith will discuss, goes into the specific equipment associated with those unit operations. And then chapter 4 looks at the different analytical technologies associated with cellular manufacturing processes. So to look at chapter 2, the guiding principles. So as with anything, everything starts with the process. So when you look at the process, excuse me, when you consider the process, we're still looking at similar aspects that you would be used to in general terms for any process, any pharmaceutical manufacturing process. Critical material attributes, critical process parameters, critical quality attributes. These are all critical to ensuring efficient manufacturing and the quality of the end product. But there's a lot of differences and challenges associated with this that are quite novel to this industry compared to other modalities, not least because we're talking about cellular products that are derived potentially from patients if we're looking at autologous therapies. And therefore we're looking, if we focus on autologous therapies for a minute, you're deriving your initial starter material from a patient who is sick with the illness that you're trying to treat, and therefore the fact that it is derived from a patient means that there is a significant level of variability from batch to batch because it's person to person. And then secondly, the quality of the cellular product that is being used as the starting material to produce the therapy is also variable because it depends how sick the patient is. The viability of that material depends on how sick the patient is. And then looking at the different aspects associated with the process, really this comes down to there's a lot of risk assessment and risk management which are possibly not included, which are different in consideration to the other biological modalities. And also the last bullet point, which is highlighted there around cost analysis, is also a difference for this modality, because if we're talking about relatively small scale facilities, and again, if we look at autologous as an example, this centralized manufacturing facility approaches has a greatly different cost model to a centralized manufacturing facility for something like a monoclonal antibody. So you have a facility which costs a similar amount to build and run, if not more to run, and you have a facility which is less automated, as the industry stands at the moment, more and has a much lower throughput. Therefore, the cost of each therapy is considerably higher, particularly when you think about the fact that you're producing a therapy which is a 1 or 2 dose therapy to hopefully cure the patient, whereas with a monoclonal antibody, you're developing a significantly higher number of therapies per batch, but also a therapy that has an ongoing treatment to a patient. So one of the big issues in the industry at the moment is the cost of the therapy to the patient. And in order to rectify that, in order to bring that cost down, the cost of goods as a whole needs to be looked at in order to reduce that cost. And that involves looking at different ways of carrying out the process, involving which is more efficient, involves less people, is more automated, and also looking at different manufacturing models, such as decentralized manufacturing compared to the main centralized manufacturing model. So this is a brief overview. So this is the bullet points on the right give an overview of the different regulatory frameworks, different regulatory considerations, and different guides that are applicable to somebody who is looking to develop a process or a facility for a cellular manufacturing process. And the diagram on the right is taken from ICHQ9, so it's not a new approach, but it's a very critical approach to this aspect, this particular industry, because of all the differences and to some degree the lack of harmonization in the approaches and the differences in the wide range of different strategies and manufacturing process design that is in the industry at the moment. And therefore, that lack of harmonization means that everybody needs to focus on this very critically from their own perspective, and there's less opportunity to leverage from other industry best practices. And therefore, as I said at the start, part of developing this guide and part of the ISPE ATMP community of practice is an aim to move towards that harmonization, to allow companies to better interact and better leverage learnings from each other, so that greater level of harmonization can improve the quality and make risk management more efficient. So one of the key factors here is around contamination control. And one of the big aspects, this over the course of the last 8 years, facility design considerations have changed markedly from the initial approvals through to the latest trends and technologies. So contamination control is critical because you're talking about, not least because you're talking about an aseptic manufacturing drug process, which is not sterile filtered and not terminally sterilized. and therefore you are looking at aseptic technique in high-grade environments for end-to-end manufacturing, and therefore the contamination control is critical. And with the essence on speed to market and the early commercial approvals, the manufacturing processes were quite open, quite manual, and very labor intensive, and those aspects are not really conducive with efficient aseptic operation, and therefore facility designs needed to address that. Small facilities with a lot of small segregated rooms, a lot of grade B environments, with operating in grade A biosafety cabinets. One of the challenges being, or the assumed challenges being, that because you're working with small bench scale operations, tactility of isolated technology may not or was not there to allow you to perform these delicate operations within an isolator. So that led to a higher grade clean room environments and operating within a BSC. So this is not desirable from a sustainability perspective. It's not desirable from an operability perspective. It's not desirable from a cost perspective. And therefore, there's been a lot of work in the process technology development to allow this to become more efficient. So as the industry has progressed, as we have moved towards more automated and closed systems, this has allowed the clean room backgrounds to be reduced. And one of the good discussions in the guide is around isolators versus BSCs. So obviously with a BSC, if you're doing an aseptic manufacturing process, you have to have a grade B clean room background. If you are operating within an isolator and it is defined under Annex 1 as a closed isolator system, which it would be in this for this type of process, then that means that you can operate potentially with a grade D background, which makes the operator's operation much more streamlined. Reducing the clean room grade means that you have much less risk of an environmental incident taking the clean room down and interrupting production, which again is more critical for cellular therapies because of the nature, particularly with autologous therapies, because you're dealing with material that is coming from a patient that is waiting a prolonged period potentially to be scheduled for their therapy. So downtime of production is very significant issue, and as such facility designs are really looked at 100% uptime or as close to as possible to ensure that there's no even maintenance downtime that interrupts that production schedule. So with all of these considerations and moving towards those more closed, automated, integrated, has led from large segregated facilities with high-grade clean rooms to more streamlined facilities which with smaller footprints, more open ballrooms and lower clean room grades, which makes things much more efficient, but also, as I mentioned before, around the cost of goods. It makes the cost of manufacturing reduced. And as that future step, when we look towards the roboticized systems that will assist with doing this, with improving that even further. So just want to look at the automation section now then. So as I mentioned, the initial therapies being very manual, being that individual unit operations required manual transfer between the different unit operations, and having 2 operators per station to ensure that everything was done and checked properly, because this is what is required through good aseptic technique. all of that is very onerous. All of that is very potentially prone to errors, and therefore, the more automated the system can be, the process can be, and the more removed from these manual operations, the better from a contamination control perspective, the better from a quality perspective, the better from an efficiency, and the better from a cost perspective. So as we looking to move towards the progression of automation has gone from manual to automated individual unit operations to automated and integrated platforms such as the Miltenyi Prodigy, to just give an example of one, and then moving towards the likes of Multiply Labs and Solaris, which are automated, integrated, roboticized platforms that are essentially their own self-contained units. And all of one of the really critical aspects of cell therapy, particularly autologous cell therapy, is the chain of identity and the track and trace of the patient material. So once that patient once the apheresis is drawn, from that point, the material has to be very critically tracked to ensure that it is monitored the whole way through the logistics of transferring to a production facility, moving through production, and then moving back to infusion into the patient, to ensure that every step of that operation is known, recorded, auditable, and that what goes back to the patient is their material and not somebody else's. And in a facility design where your production capacity is increased through scaling out rather than scaling up, and you potentially have a lot of different unit operations operating in parallel, a lot of different batches being manufactured in parallel in the same room, then that becomes very critical, because mix-ups can potentially be easy, and that is a big challenge for the industry. So having your MES properly set up, and everything, the chain of identity properly linked into each manufacturing step is critical to ensuring that this is your chain of identity is as robust as possible. And this slide just gives an overview of the proposed automated digital architecture, with everything centering around the manufacturing execution system in your CMMS, with all the different aspects associated with the facility linked into that, so everything can be tracked and monitored and accessed potentially in real time, and everything is available in one area, and everything is recorded together, so you're not needing to look at different different automation platforms to be able to get to try and get different pieces of information. And one of the aspects that I was mentioning before was cost of goods for autologous therapies in particular will potentially push us to the decentralized manufacturing model. And for a decentralized manufacturing module, the digital strategy is critical, because all of the individual, you will end up in a situation where you have a lot of small modular manufacturing operations, which are all controlled essentially by one centralized function. And that centralized function is where you have your quality risk management, your quality procedures, your manufacturing procedures. Everything needs to be managed through that central system. And there needs to be a central focus on everything that's being released from all of the different manufacturing operations. So having an efficient, effective, streamlined digital strategy that will allow you to see all of that information in real time will be critical to an effective decentralized manufacturing strategy. And so this, the last slide that I have is just on the technology roadmap. So this just gives an overview of all different things that have been through, that I've talked about with respect to how the industry started and moving towards where the industry is going. So we started at section 1 there, which is the very open and segregated manufacturing approach. We're now at stage 3, which is looking at the robotic systems are becoming more prevalent. They're still in the somewhat discovery phase to actually get them into an efficient way of manufacturing, but we are in that kind of that stage 3 area at the moment, and future looking is where we would like to get, which is to the stage 4, which is where we're focused more at minimization in manual intervention, moving towards lights out as far as is practical, real-time release using automation and highly automated digitalized facilities. So that's an overview of section 2. I will now hand over to Judith to discuss chapter 3.
I think, Stephen, we have a question on the automation part. Maybe before we continue, we can take that one. Is this automation streamlined applicable to allogeneic therapy production? I think maybe if you would just go back to the ecosystem slide. I think from my perspective, Stephen, feel free to disagree. We would probably not have the patient orchestration tool. At least that's what I usually see. There are still functionalities in the orchestration tools that we can use when it comes to really specific treatment slots, also on an allogeneic appointment. But because we usually produce to stock instead of really produce to patient, usually I would say patient orchestration would be excluded. Other than that, from my perspective, I would say it's very much the same. Do you think?
Yeah, I would agree. I mean, you have a difference in your patient orchestration is obviously critical is more to the autologous, because the starting material is coming from the patient. You would have some level of scheduling with respect to allogeneic, because you still need to get donor material, but it wouldn't be. It would be more of a, as you say, it would be a stock replenishment rather than a direct kind of link into production scheduling as such. And I think that I agree that this would then be very much the same. And when it comes to the kind of discussions around decentralized versus centralized, that kind of thing would be with respect to autologous. With respect to allogeneic, it would depend on the process and depend, because some allogeneic processes would still be relatively small scale, and donor batches would be relatively small for each donor harvest, and therefore you'd still be looking at that possibly decentralized model. And then other different types of autologous processes would be able to operate in larger scales. I think the largest that I've seen is still at the kind of 1,000-2,000 liter bioreactor scale. So those types of allogeneic processes could be more suited to a centralized facility.
There's another question, Stephen. Any laboratory information management system that you would deem as most suitable for decentralized manufacturing system, like you mentioned?
Laboratory information management systems are not my area of expertise, so that maybe Judith can speak to that.
Absolutely. So I think, honestly speaking, when it comes to LIMS system as a whole, so first of all, Stephen and I both agreed before the webinar, we do not want to provide any vendor names in terms of recommendations. I think I hope we have your understanding if we do the same here. But when it comes to considerations, I think what we have definitely seen in the past is that some of the traditional LIMS systems are not flexible enough, or do not support the kind of flexibility and the kind of integration and interconnection that we really see for cell therapy processes. Just as one example that we see quite often, if we manufacture to strength, and that means that we can have between 5 and 7 expansion days before we harvest, and at the same time we need a cell count and a flow cytometer analysis every process day. That requires flexibility. This is shared flexibility between both manufacturing and our QC personnel. That is something that sometimes is really a challenge when it comes to traditional LIMS systems. So I think when it comes to considerations, I would definitely recommend looking into the level of flexibility that is required, and also the level of integration between the LIMS, the MES, and the ERP system, and really reconsidering about which system is doing what and which one is in their lead. And what we have seen as most successful is if the MES knows the calculations, it knows how many expansion days we need, it can be the leading system when it comes to orchestration of the sampling. That doesn't mean that the LIMS can't create, for example, the sample IDs and also the sample labels. But really thinking about this tighter integration between the two systems is definitely something that we have seen as quite useful. When it comes to decentralized manufacturing, honestly, same thing. Usually we would see LIMS systems being on an enterprise level with a side-by-side implementation, and then really on a batch perspective, we can use the site-specific LIMS system, but an enterprise orchestration is definitely helpful when it comes to decentralized approaches.
Yeah.
Awesome, thank you. Happy
Yeah,
to.
thanks, Judith. Yeah, so please continue.
Awesome. All right, so then I will share the screen. I hope you can see it.
Yes.
Excellent. All right, so then, as Stephen already mentioned, in chapter 2, we really took this approach of how to do things, what are best practices, how to consider our equipment and process development as a whole. And in chapter 3, we went further into details of the actual production technology. So again, as Stephen already introduced, we have been focusing on both autologous and allogeneic. And in our chapter 3, we really went into the individual steps of our process. And we did not create one for autologous and one for allogeneic, but really looked at the individual process steps and then discussed a little bit around what to consider, what are the things that we need to look out for, rather than really making suggestions, more into what are the considerations that we see here in these particular processes. So I assume because we are meeting here on ATMPs, we don't need to discuss too much about the actual process, but maybe just very briefly, we have now just assumed that we are starting with frozen cells that are arriving from the hospital. And then we would probably have a storage. And then, of course, we need to thaw our cells first. And then we connected our prep, enrichment, and selection section. So where we would, for example, select for CD4, CD8 cells, or enrich CD4 and CD8 cells, or a particular phenotype of NK cells, or whatever we really need. And then we would go into an activation step, because that's what we have been discussing usually then on the next process day, that we activate our cells usually with beads to make sure that they, again, have the phenotype that we really require. Afterwards, we would consider a genetic modification step where, of course, we have different possibilities, different delivery routes. We will come to that in a moment, to create the target cells based on the cells that we now have. And then afterwards, of course, our cell expansion, where now we have usually between 6 and 7 days. Maybe today we are getting into shorter cycles, where we are now expanding the cells as required, and of course analyzing what we have and if we have already reached the strength that we need in the end. Then afterwards we would harvest the cells that we need. The debeading process from our perspective is really important. So basically the beads that we have been using at the beginning, making sure that they are now removed. And then we would formulate our cells to the required strength, fill them into vials or bags, And in the end, we have also included the inspection, the visual inspection phase, and then of course labeling, and then we would freeze our cells, probably store them in a liquid nitrogen tank or something similar, and then again use them for shipment. So if we think about this being our more or less standard autologeless process, then really the main difference that we have considered for the allogeneic cell therapy is that we can perform cell banking. Obviously, that's not always done, but honestly speaking, when we have had this discussion, we realized that pretty much everyone who can do it in their process really does cell banking. So that's why we also included a section around formulating our working cell banks or master cell banks. And then based on that, of course, filling them and labeling them, another cryopreservation step. And then afterwards, we would thaw that and go into the process. The main difference that we try to indicate here is, of course, the scale. As Stephen already mentioned, here we are talking more about 200 milliliter cultures. Here we are talking maybe 1,000 liters, something around those lines. Honestly, I think we also see a lot of development right now when it comes to the actual scale up in the allogeneic space. So I think a lot of the equipment vendors really provide small scale bioreactors and basically provide us with the promise for a scale up rather than actually already having a lot of experience, at least from our group. So this is really where we see right now the scale of around 1,000 liter cartridge. And of course, that also allows for more automation in individual steps. For example, when it comes to the filling, we definitely have seen that more automatic filling lines are used for the allogeneic space. Same for inspection. But we can go into the details a little bit later. So if we think about our process, and we just ran through it, there are a few considerations that we really wanted to highlight. For example, when it comes to the leukapheresis thawing, obviously we would usually receive or store our frozen cells. Now we have to thaw them, and mainly there have been two ways that we have been discussing. One is the more traditional and a little bit less controlled way, where we are really putting our cells into a water bath or in a controlled environment. That is still done. It's not that we have not found anyone who would say that's the right process, but the majority of our discussions went around really controlled thawing processes where we are using equipment that in a controlled way increase the temperature by a certain amount of degree over a certain amount of time. Considerations that we really found here were mainly around places and around the possibility to create protocols. So between the different vendors, I mean, there are not too many, but between the different vendors who provide a solution like that, that's really the main difference and the main consideration that we saw as to, can we combine bags and vials? Can we only use vials? Do we have maybe an automated way to select the places to put the vials? And then, of course, the protocol itself, like the amount of degree per minute, that's more or less a given in this kind of equipment, but then there are additional things that we really can receive. For example, the amount of reporting of timestamps, for example, between the start of the thawing cycle and the end of the thawing cycle, using different parameters that the system recognizes itself. So that's really where we have seen considerations that make a difference if we select the vendor from these tabletop equipment. And we also included in the same section cell washing. Again, you probably all are familiar with the process. So we know that cell washing happens in a lot of places during our process, but we just included it in the first step. And then, of course, every time that it would reappear over the process, the same would apply. Now, for cell washing, we have mainly distinguished between the two methods. So counterflow centrifugation on the one side and spinning membrane filtration on the other side. in comparison with classic centrifugation. And what we have found is that it's really a lot about the sensitivity about the cells that should define our cell separation process. A second parameter that we have discussed a lot is how good can our equipment separate between living cells and dead cells, because what we really want is, of course, our living cells separated from the old media, our freezing media, and any dead cells. So really the precision with which the separation between living and dead cells happens, depending on the leaf force, that is something that we have seen as a consideration. And then also, let's say, the level of sophistication. So between an equipment that allows us to do one washing cycle and equipment that allows us to use different washing solutions and different washing cycles, really, we are back on the program definition. That is something that we have seen with the cell washing process. Of course, there are also possibilities to include the cell washing in a cell processor, but I will come to that in a moment. More or less, we are really seeing our cell washing cycle as tabletop equipment. And the next one, as I said, enrichment and depletion. So, for example, the classic CD4, CD8, where we are trying to really enrich our cells with beads, and with that influence the phenotype, but also influence the cell composition when it comes to, for example, depleting particular cells. Here, of course, we are talking about the fact that we are using beads. From an execution and equipment perspective, there are definitely different ways how to do that, specifically, again, when it comes to the protocol definitions, but also when it comes to the sensitivity of the method. Of course, we have the possibility to do that completely manually. We go into our isolator, the part that Stephen already discussed, add our beads, perform any shaking process, and then let them incubate. That's definitely a possibility. But when it comes to really adding the cells using a specialized equipment, that's where we have seen differences in the sensitivity as well as in the composition. Then we have the activation step, again, where we want to, for example, promote a particular cell phenotype. Here we mainly talk about the fact that we are adding growth factors, interleukins, maybe antigen-presenting cells to our cells. This is the first step that we usually see if we are going for an automatic or semi-automatic cell processor, that this could be done using one of the modules, so that we basically already feed our cells. Maybe taking this example here, if this is our cell chamber with a steering tank, and we add our cells into our bioreactor, we could actually add using one of the ports our activation ingredients. Here, a consideration that we have very much seen is, of course, a cost factor, because, for example, when it comes to growth factors, they are really one of the biggest cost factors when it comes to our material. So definitely important as to the volume with which we want to work, and of course the volume being dependent on the bioreactor or vessel that we are using to incubate the cells. Other than that, we definitely see, if we're not using the module of a cell processor, that this can be done in a manual step using the isolator and then putting them back into the incubation. Then as a next step, we would have our lean modification. So obviously we don't always have that. For example, in the allogeneic space, of course, we have processes that are not genetically modified, but here we really went for the full program, including genetic modification. And so what we have discussed here is, of course, first of all, it's dependent on the delivery method, and we have separated between viral and non-viral transduction. First of all, I think that really made a lot of sense. And where we said, when it comes to viral modification, obviously we are adding our lentivirus or maybe adenovirus to our cells, which, very similar to the activation, can really be either a module in a cell processor or a manual step. Something that we suggest to consider here is really the addition rate, because that is, and also the shaking of the vessel during the addition of the vector, because that again has been shown in the past and also published, really changing both the cytotoxicity but also the success rate of our viral transduction. And what we have seen here is that if we do that with a manual process, of course, it's very much about the SOP and the experienced operators who really know exactly how to do that to receive the best success rates. When it comes to non-viral modifications, again, we have discussed a few of the different programs. For example, when it comes to electroporation, of course, we have the dedicated tabletop equipment that we use for our electroporation. And we have made a few discussions around what really makes a difference there, but the basic parameters we have found are pretty much always the same. Again, it's more a matter of the volume that we are working in. And then, of course, again, we have modules for our cell processor. And then we also have our microbubbles method and sonification. I think that's a little bit less used as of right now in the different processes. But we also have some discussions around those lines, pretty much comparing different equipment that you could use for that. But as I said, the basic parameters would usually be the same. And then the last aspect for genetic modification, we have considered chemical transfection methods. And then, of course, we have semi-automatic processes, but also our manual execution, where we just use our hands and the VSC or isolator, and all is done manually. The expansion is where we have put most effort in. And we also see here this diagram on the right side. We have differentiated between suspension and adherent cells. And then, of course, here we would have our T flasks or cell stack, depending on the volume. And then we also have, when it comes to multi-layers, our fixed bed bioreactors or microfluids. And then on the suspension cell equipment, of course, we have our Erlenmeyer flask in different shapes and sizes, where we have gas transfer, our gas permeable cell bag, and then, of course, the wave rocker, where we really just provide a substance for our cell bag mostly to perform a rocking process that would usually be done, for example, by operators, but in a more controlled way. And then, of course, a single-use bioreactor, where we have really a programmable piece of equipment that allows us to, for example, select the program over the next days. And here we are usually seeing single-use kits. Main consideration here that we made, one that we found really important, is the cell phenotype. So the NIH published, I think 2 years ago or something, a very interesting publication about the difference between cell bags, flasks, and single-use bioreactors, and how the selection of the equipment actually influences the cell phenotype. And of course, the cell phenotype, so for in the example, the maturity of the T cells, very much affected their therapeutic treatment and their therapeutic success. So I think that's one of the examples where maybe we as an industry are not considering something strongly enough, and then we realize, oh, that really makes a huge difference. And in their publication, they suggested that it might be hypoxic conditions that are introduced by the bioreactor. So that's something where we felt it's really important that we put emphasis on this, because it really makes a huge difference when it comes to using our process. Another thing that we really feel is very important when it comes to the bioreactors particularly is the measurement of metabolites. So I think the most processes that have been accepted for approval over the last years really go for a daily cell count. We measure the viability using the classic cell counters. But of course, now we also have bioreactors that do inline measurement of metabolites, and with that we can improve and increase our process control. At the same time, this can't necessarily really replace the cell count if we have filed our approval with a cell count process. So that's another consideration that we really suggest, particularly when it comes to the development of new processes. And that's definitely not news to anyone, but we have also included a little bit of a discussion as to, let's select first really what we want to use also for scale up. Do we want to focus on scale up or scale out, as well as, of course, GXP considerations, because changing the method, particularly between those, but also when it comes to between bioreactors, is just really a challenge in our industry because of cell variation and, of course, small starting materials. The rest of them, I think I can probably summarize. I'm also checking the time here. So harvesting obviously is pretty much a cell separator. The bead removal is also pretty much the cell separator. And when it comes to the filling process, that's really the part where we have discussed a little bit about how much can you automate, particularly when it comes to larger scales. So, for example, in the allogeneic space, there are definitely a few solutions that allow us to do automatic filling. Here, as well as with the visual inspection, a few considerations really when it comes to selection between bags and vials. Some of the equipment allows only for one, other equipment allows for both, and also provides different levels of flexibility and different levels of combination. So this is really where we have seen a very high focus on selection based on that. And then cryopreservation, just going to the next slide here. Obviously, here we really have the different styles when it comes to, do we use nitrogen or do we use electric plate freezers? There is definitely, of course, the advantage in the electric plate freezers when it comes to necessity of nitrogen, of liquid nitrogen, but also, for example, what happens if the flow of nitrogen into our top loader or front loader is interrupted, particularly with our autologous one chance processes. This is definitely a risk that we need to consider. At the same time, of course, there are also aspects that really speak for the liquid nitrogen tank, but we need to find the balance between how much risk do we want to take, particularly in this feeding protocol of our liquid nitrogen into the tank. So before I come to analytic technologies, any questions to the last clip there?
There's no question in the chat, Judith.
Awesome, perfect, thank you. So then I would like to just wrap it up with analytics. And of course, again, we just went through the complete process and discussed a little bit about what do we need to do here. And first of all, so we went through all of the approvals, but also our own experiences, process development, and looked at, first of all, what do we need to analyze? Over our process, obviously, we would need usually the flow cytometer to recognize our target cells. Of course, there are other methods, but this is really something where we see the most focus on the flow. And then cell count and viability, and as I just said, also the consideration about how much emphasis do we want to put on the metabolites. Then, of course, we have our sterility at different parts of the process. Of course, it's not usually done in 6 days, but really in a combination of different days. Obviously, a little bit dependent on our release time and how much time do we have after the production of our cells, if we need the sterility results on the same day or not. So based on that, of course, the question around the sterility. And then our classic release test when it comes to mycoplasma, adherence, and the toxins, and so on. And if we go into the release test just a little bit more in detail, we have separated between our classic T-cell therapies, which like CARs and TILs on the one side, and then the stem cell-based therapies on the other side. as we see, the majority of them is very similar, right? So when it comes to sterility, the BACT, mycoplasma, that's all very common. The biggest difference is really when it comes to the identity and how we analyze the identity of the cells, because on the left side, of course, we would usually go for the composition of the cells. Do they have the particular surface proteins that we need? Yes or no. It also depends a little bit, of course, on how many target proteins, for example, we have when it comes to multi-genetic modification. It's probably better to automate more in comparison to having one surface protein for transduction. And then on the right side, it's really more about the phenotypes, the cell cycle analysis, and differentiation tests and aspects around that. Obviously, this again is not extensive. It's really just a combination of which tests we really see. And a few last comments, what we also see as very important here. One is our typical IPC and QC tests by product type and process steps. So that, of course, we really have very early on a complete testing plan when it comes to our release tests. Ensuring identity, potency, purity, and safety, the usual. But also when it comes to how do we have the possibility, for example, to combine our identity tests. So that's really what all of these discussions are around. And then a few general considerations here. For example, when it comes to Pharma 4.0, where unfortunately, specifically in the cell therapy process, we are still a little bit behind when it comes to digitization. We already had the LIMS discussion. This is just something where we have really put some emphasis on, because we feel it would really be a shame if we have the possibility to automate and speed up our manufacturing process, but QC and IPCs are behind, specifically again with this very, very tight connection between manufacturing and analysis. This is really something where we feel we need to make sure that we also put emphasis on automating and improving the data connectivity from our analytics department. So yeah, I think that was pretty much what we wanted to share. Just definitely our acknowledgements, because again, that's not Stephen's and my work. It's been the work of a huge group of people and an amazing group as this, because we really had a lot of amazing discussions, and it was a lot of work, but also a lot of fun to come together and have all of these discussions. I think we have about 1 minute if there are any last minute questions.
Yeah, thanks, Judith, and thanks, Stephen. Any questions from the audience? Any burning questions? So maybe one final question from my side. It's a complex one. I was just referring to your comments, Stephen, in terms of driving down the cost of goods. You're alluding to decentralized manufacturing as the way to go for the future. But I tend to think this is a very complex topic, right? I think it also requires, also tying into Judith, your part of the presentation, a lot of analytical enablement that's really required, right? So what is your view on this? What level of technology maturity this industry is really in to enable that?
So it is a difficult question to answer, because really, in order to properly understand or to properly realize the savings associated with decentralized manufacturing, the cost of manufacturing, the cost of deploying your manufacturing module needs to be very much reduced than what you've done, a comparable consideration for constructing a facility. So purely from a facility design and operation perspective, you need to have a standardized module that can be deployed wherever you need it to be deployed. So you have one design and you just replicate it wherever you go. In order to keep the cost of the actual manufacturing down, that needs to be largely possibly a discussion can be had around the modularization of that facility, whether it's an off-site full module that gets deployed somewhere, or whether it's built in modular sections. That's another consideration that needs to be looked at from a cost perspective, and also then the method of repeatability to ensure that you have comparability between the different areas. So if you look at the more roboticized systems that are on the market, I think these are more expensive from a technology perspective, but they're also going to lead to more repeatability because because of the way that they operate. But then there may be a cost consideration that they're not practical to deploy in all of these individual modules because the cost may be too prohibitive. So it may be a case of looking at more standard integrated automated platforms than maybe robotic systems. So there's a lot, from a technology point of view, there's a lot of consideration to be able to look at what really is the most cost-effective model in order to be able to do that effectively. It's certainly not a straightforward answer to the question. And that the cost in different geographical regions, the cost of also comes into that, and the availability of the cost of energy, the availability of green energy, and all that sort of thing. So there's a lot of environmental considerations and locational considerations that come into that as well. But and then I'll let Judith talk about the digitalization piece.
Yeah. So,
yeah,
but
I think that's really.
I think it's a very comprehensive answer already. We are over time. I just wanted to still entertain the last question in the chat, right? So Judith, maybe give us a very concise answer. Do you see rapid sterility testing being more implemented in ATMP release?
Yes. Yes, for sure. We can really keep it that short. I mean, obviously, that's just one of the topics, right? Together with improving our release cycles and other considerations how to improve it. But yes, we definitely see the tendency from my perspective.
Okay. Yeah, thank you very much. Once again, thanks everybody for staying with us. Hopefully this is a very informative session for all of you. And again, thank you, Judith, and thanks, Stephen, for taking the time to share your knowledge. This is actually the first of several ISPE ATMP Good Practice Guide presentations. Do stay with us. We'll broadcast upcoming webinars in time. By the way, my name is Peijing Zhang. I forgot to introduce myself at the beginning. I lead our APAC ATMP COP. So for those who are interested to view, always check our ISPE Singapore affiliate YouTube channel to watch the recording, and look forward to seeing all of you in upcoming webinars. Thank you very much. Have a good day, everyone. Thank you.
Thanks for having us. Bye.
Thank you. Bye.
Bye.
Thank you.